Okay, excellent. So thank you very much, Francisco, for inviting me to talk at the symposium. There's been some fascinating stuff already, and looking ahead, I can see there's some nice talks tomorrow. So thank you for arranging this. So today I would like to talk about some work that's a few years old now, but I think it's of relevance given the talks that we've already seen today, in that it particularly talks about whether size matters, in the sense that whether some of these small-scale structures we've seen, like eddies and filaments, whether the biological dynamics associated with that matter at the large scale. So this is work, most of it was done with Marina Levy, based in Paris, but there were also a number of people at NOC or previously at NOC who I've marked on this slide, who have also been involved in this work and deserve credit for it. So this is something that has already been shown. This is essentially a quick schematic of the biological carbon pump. And so often when we're looking at biogeochemical cycles, particularly in the Earth system, then these are the sort of things that motivate it in terms of how the organic material gets processed, how atmospheric carbon dioxide gets taken up in the ocean, converted into new phytoplankton, and then what happens thereafter. And people have used global models, Earth system models, to estimate the size of this flux, the biological carbon pump, and also how it changes into the future. One of the things which is usually known but not always acknowledged fully is the fact that variability in the ocean goes all the way from the basin scale down to the millimeter scale, indeed down to the scale of the organism, which may be a few microns in size. And so this is a nice little sequence of satellite images. This is just off the northwest coast of Australia. It's showing you a proxy for chlorophyll abundance. So the paler an area, the higher the concentration. And what you can see is this constant churning by the oceans, but also there is a very strong spatial variability. Wherever you look, you see these strong gradients. And so at the top, you've got this scale of 1,000 kilometers. And what we know is that a lot of the global models that we use typically can only resolve things down to 100 kilometers if they're doing quite well. Whereas we know from satellite images like this that there are strong spatial gradients going all the way down to, you know, a kilometer, if not less. And that's something that Elisa showed nicely in the work that she was talking about earlier. If we look at in situ observations such as these are just two transects of phytoplankton and nitrate abundance. Along the bottom is distance. And you can see that on the scale of just a few kilometers, you can have 2 or 3 factor changes in the abundance of these, but also that they don't always seem to be that well correlated. So we need to start thinking about whether these small scales matter. If we look at it from the point of view of modeling, we need to have equations that describe this. And so what will be familiar to a lot of you will be the equation here, which is just the equation describing how a tracer moves. So you have the rate of change of tracer at a point is given by the first term on the rate h side is the advection by the currents, and then the second part is the interactions. And this was illustrated nicely in the first talk today in terms of the different components influencing tracers. So for the phytoplankton, those terms might be primary production and grazing. For nitrate, they might be remineralization and uptake. But the thing to stress is that more often than not, they are non-linear. So once again, that was very nicely explained in the first talk today. A trick that's often done is to split the current and the tracer into a mean and a perturbed component. And so if you do this, which is the equation I've shown on the bottom here, you see that you pick up these different terms. So if you have a global model which can only resolve down to say 100 kilometers, then that means that you're only getting the first, third and the fifth terms here. You're not getting the second and the fifth ones. The second one is something which has already been highlighted by Elisa, which is that things like eddies and filaments can play a strong role in advecting material. And so this extra term is often corrected for in models using things like a Gentwick-Williams parameterization. Still far from perfect, but it's a way of at least trying to capture those processes in global models. The fourth term, however, is almost universally ignored. And this comes from the fact that if you have nonlinear interactions, if you split your field into a mean and a perturbation, you pick up extra interactions between the perturbations. And so effectively you have the nonlinear interactions between the fields at the small scales as a contributing factor to what happens at the large scales. The question is it significant? Is that term large? So the way I'm going to talk about it is a two-pronged approach. First of all, I'm going to show you a model. And as you'll know, the benefits of a model are that you know everything. The downside of a model is that you also know it's almost certainly wrong. You know, every model is an approximation. So I'll then turn to observations, and here we know very little in terms of we have little data, but we know at least it's reflecting the true system. So what I'm going to do is to take a relatively simple model, and this will look familiar to Louise in terms of it's a nice simple reproduction of a double gyre, slightly tilted, so it's slightly different. And this was a simple model set up by the French group a number of years ago, just for looking at biogeochemical questions in a more simplified setting. And so you have a jet going across the center of this box with a subpolar gyre above, subtropical below, and very crudely it coincides with sort of the structure of the North Atlantic. And the advantage is that they ran this model at a range of different resolutions, all the way from 2 kilometers up to the order of a degree. The forcing is exactly the same for all of those runs. So this means we can actually quantify how important the small scales are. So this is just to show you how this model performs. This is showing you chlorophyll, and you can see the takeoff in those bright red colors of the North Atlantic bloom taking off within the model. This has just been superimposed on the North Atlantic to give you an idea of what sort of geographical area it might correspond to. It's not supposed to be exact. So how do we calculate the importance of those eddy reactions? What we do in this case is to take our full field at the highest resolution, so that's shown on the left here for phytoplankton, that's concentration, and then we run a 2-degree filter over it to remove a lot of that small scale variability. If we then subtract the filtered field from the full field, that gives us the interactions at the small scale. One of the first things we find is that there is a scale dependence. So each of the lines on this graph corresponds to one of the main processes taking place within a biogeochemical model. So NP is the uptake of nutrients by phytoplankton, RP is the regeneration of nutrients, GRP is grazing by phytoplankton, GR is grazing of zooplankton on detritus, and then MZ is the mortality of zooplankton. And this shows the relative magnitude of that process in the model as a function of the model resolution. And you can see the model goes from 1/54th of a degree at the bottom to 2 degrees. And you can see that in all of those processes, they are sensitive to the resolution of the model. What's perhaps a little disconcerting is that some of those increase with resolution and some of those decrease in resolution. If you broadly define the sub-mesoscale as between 2 and 30 kilometers and the mesoscale as above 30, then you see that the contribution to the total, if you look at the highest resolution model, comes about 1/3 from the sub-mesoscale and 2/3 from the mesoscale. Bear in mind that most global models don't even capture the mesoscale accurately, and so potentially there is a lot of this contribution being missed. If we look at the particular fields, then we start to see a more complicated picture. So this is phytoplankton, and it is showing you the contributions to the rate of change of phytoplankton as a function of latitude in the model, but broken into the different components of that equation at the start. And so in the top, in red, this is the terms due to advection, the total, the mean, and then the eddy flux. And then we have blue vertical mixing, and then in green, we have the biogeochemical reactions, the mean and the eddy reaction. So if you look for the phytoplankton, then you see that the eddy reaction for the phytoplankton is actually the same order of magnitude as any of the other fluxes. It is an order 1 term. And so for phytoplankton, the evidence from this model at least is that the eddy reactions are very important. If we look at the same for nitrate, I apologize for the dashed line being blue rather than green, but you see that it's actually much smaller. And so the implication here is that the eddy reaction terms don't matter for nitrate. So we seem to have somewhat contrasting results from the model. So the next question to ask is, what do we see in the observations? And so the idea is that we try and use exactly the same equations that we use in the model for calculating these interactions, but rather than calculate the phytoplankton and the nutrient concentrations in the model, we take those concentrations from reality. And so we do that to calculate the ratio of the eddy to the mean reactions. That tells us how important these small scales are to the larger. And just because it's the only things we have data for, we have to focus on primary production, grazing, and if you're interested at the end, I can tell you about mortality as well. You've seen this before. This is showing you abundance of phytoplankton and nitrate. This was actually a spatial survey. It's been stretched out to show you the variability. And so we have this data that we're going to use to estimate the eddy reaction terms. And what we do is to simply use those observations and feed them into the equation in the model. So this is the equation, maybe familiar to a lot of you. It's a Michaelis-Menten type equation for the uptake of nitrate by phytoplankton. We just plug those observations in here to calculate the uptake term. So on the left here, this is latitude and longitude. It shows you the track over which the data was collected. It was done over a grid, or tried to, weather intervened. In the middle is the correlation between nitrate and chlorophyll. You can see they're actually rather weakly correlated, which is an entirely different but very interesting question. And then on the right, this is a graph of how the eddy reaction varies. The ratio of the eddy reaction to the mean reaction varies as a function of the half saturation for nitrate uptake. You can see that there is some variation, but the overall result is that the eddy reaction is less than 4% of the mean reaction. It's actually rather small. If we do the same with grazing, for here we have two data sets in red and black. This is once again showing you the data as a transect. We have a similar term for the grazing, which is a Michaelis-Menten dependent on phytoplankton. Once again, a half saturation, which is governing the rate of uptake. And here, in analogy to the phytoplankton slide, on the left, showing you in red and black the paths of the two surveys. In the center, you can see the lack of a correlation between chlorophyll and zooplankton, or at least a very weak one. And then on the right is the ratio of the eddy reaction to the mean reaction. And this time it's even smaller, less than 3%. So observations then seem to be saying something different in that at the moment they gives us, rather small answers, but these examples that I've given you from the observations have been done using local surveys. And so, you could argue that we're actually getting a very distorted picture because we're only looking at two places in the ocean, and that at a particular time. And so obviously we want to look at how things change with time and location, and an obvious thing to turn to if you're looking for observations is satellite data, which potentially can give us the information we need globally every couple of days. The catch is that we cannot currently get nitrate and zooplankton abundance at a similar resolution. Nevertheless, we can still put an estimate, an upper limit on the impact of these small scales to the larger scale. So if you look at and think a little about the mathematics of the ratio of the eddy reaction to the mean reaction, you can see that it is actually comprised of the correlation between the two fields, in this case phytoplankton and zooplankton, multiplied by the coefficient of variation for phytoplankton and for zooplankton. So we know that the correlation between the two has to be less than 1. It cannot be larger. We can calculate the coefficient of variation for the phytoplankton directly from satellite, and the coefficient of variation for zooplankton, we don't really know, but we can at least try and estimate it from the data we do have, and it's likely to be the order of 1 or less. So that's what we do. We take a look at the satellite data, and what we find is that for phytoplankton calculated from the coefficient of variation calculated from the satellite estimated phytoplankton is less than 0.6. From the in situ data analysis, we found that the correlation between phytoplankton and zooplankton is quite weak. It's about -0.5, and that between phytoplankton and nitrate is -0.3. From the limited data we have, then we can calculate the coefficients of variation for zooplankton and nitrate as well as phytoplankton, and they all come about the same, about 0.3, 0.4. So putting that together, what we find is that for the grazing, the ratio of eddy to mean reactions is about 12%, and for the primary production, it's about 7%. Okay, so we now try and bring all of those together. So what I've got here is a table which is showing you the ratio of the eddy reaction to the mean reaction in all of the three cases that we've looked at it. So we have it for the satellite case, for the in situ case, and for our idealized model. I've put it together for two parameters, for primary production and for grazing. So first of all, looking at primary production, you can see that there is quite a discrepancy between the model and the observations, in that the model has a value of about 0.6. So in that case, the eddy reaction is very significant. It's about 60% of the mean reaction. Whereas in situ and satellite, it's much less important. You know, we're down at the order of 5 to 10%. For grazing, however, we find that the model gives us a smaller contribution by the eddy reaction. So now we're down to about 10%. And in fact, that's more consistent with the observations in this case. So I guess one conclusion we can draw from this is that there is no consistent conclusion to draw from this in terms of the approaches that we take, model, in situ, satellite, all seem to give us, you know, differing answers when we look at different parameters. And there's no consistent picture appearing. So what can we conclude from this? I think the first thing is that we still don't really have an answer to whether these small scale nonlinear interactions actually matter on a global scale. There is too much uncertainty in terms of we've only looked in a few places and we've only used one model to do our analysis. In one extreme, given by the model that we have, then the eddy reactions might be the same order of magnitude as the mean reactions. And so if that was the case, and I'm not saying it is, but I'm saying, you know, we have one model that shows it might be, then that would imply that our global models are missing an order 1 process from the biogeochemistry within them. So potentially a very significant issue with modeling the Earth system. And that same model itself has its own, shall we say, inconsistencies, or at least discrepancies, in the sense that the eddy reactions are important. They're order 1 for ecology. So for phytoplankton, you know, they were demonstrated to be very large, but less so important for biogeochemistry. And so for the nitrate, it was demonstrated that the eddy reaction was quite small. And so this is also telling us that the answer to our question is going to be dependent on the question we're asking. If we're interested in how nutrients are cycled around the ocean, then potentially, and I say potentially because we've got limited evidence, then these eddy reactions might not matter so much. But if we're interested in the ecology, for example, how primary production by phytoplankton might be propping up zooplankton and in turn fish, then, you know, there is limited evidence here suggesting that the eddy reactions might actually be rather important. At least on the basis of the limited observations and the single model that we have, there is an indication that the model might be overestimating the eddy reactions, in that even though there was some variability between the observations, none of them suggested that the eddy reactions were more than about 10% the size of the mean reactions, whereas the model was predicting they could be the order of 1, 50, 60%. Another issue that it's highlighted in terms of how we calculated it, the eddy reaction to mean reaction ratio in the satellite data, is that the correlations between fields can have a major influence. And so whether the phytoplankton and nutrient field are correlated or the phytoplankton and zooplankton field are correlated has a major impact on the magnitude of the eddy reaction to the mean reaction ratio. And even though this is a very easy thing to gather from models, it's still something which is very much an open question in situ. There are time series that have looked at it, but from a global perspective, the question of how strongly correlated we expect these fields to be and how that varies through time is still, I would suggest, an open question. So I guess I'd have to admit that this talk hasn't really given you much by way of answers. I mean, hopefully it's instead given you some questions that, you know, are of potential interest for your future research. So I've just tried to capture a few of them here. The first one is the obvious question, and I think it still is a pressing question from people doing global modeling, and that is it possible to use a lower resolution for reproducing biology in models than the resolution that you use for physics? So one of the suggestions that has been put forward in order to allow us to run climate models faster is that we run the physics, say, at a 10-kilometer resolution, but we run the biology, say, at 100-kilometer resolution. I would argue that at present, we don't know if we're throwing away something important in doing that, because we don't know how important the nonlinear interactions are in those processes that we're discarding. I think another interesting thing it's thrown up is whether the simplicity of our models is actually biasing our answer. So I mentioned already that the models seem to suggest that eddy reactions are a greater contributor in our model than in observations. So one of the things that I think it would be interesting to explore is whether that is a function of the complexity of the model. So the model that I used for this study was very simple. You know, it's like a lot of the models that we've heard today: nutrient, phytoplankton, zooplankton. But that means that signals get transmitted very quickly through such a single system. And it's very difficult to have the range of complex behaviors and internal dynamics that you would get in a more complex system, and indeed in reality. So there is one possibility that in reality, the greater complexity is actually damping these effects. We don't know, but it would be interesting to try the same model experiment, using a range of models of different complexity to try that. And then the final question that I want to leave you with is, I guess it's to some extent the marine science equivalent of how long is a piece of string. How many observations do we need in order to determine how important these nonlinear interactions are at these scales of kilometers or even smaller? How many observations do we need to collect? At present, we have a tiny number, but if we're going to answer this question, then at least we need to be able to make an informed estimate of how many and in how many locations. And even though that's a very difficult thing to estimate, I think that with high resolution models, it's some things that we can at least put a rough figure on. So that's it from me, and I'm happy to take any questions. Thank you.